you guys will see that pop up on your screen.
Oh yeah. Oh, went away again. Okay, is it on the big screen now?
Yeah,
perfect.
Looks great.
Cool. Okay, so hi everyone, I'm Kate Fell and I am a final year PhD student at University of Glasgow, and I'm just going to give a really brief introduction to what I've done with my PhD research. So before we start, just a wee bit about me. So I am born and raised in Glasgow, Scotland, and I've studied basically here my entire life. So I did my bachelor's in forensic investigation at Glasgow Cali. That's where I got my taste for analytical chemistry. I then went on to do a master's in pharmaceutical analysis at the University of Strathclyde, and I got to do my research project at the European Directorate for the Quality of Medicines and Healthcare in Strasbourg, France, which was really cool because that's where that's the labs that do the European Pharmacopoeia. So it was a really good opportunity. And now I am at the University of Glasgow and I'm doing my PhD in analytical environmental chemistry. So up in this, oh, I'll put the pointer on. Up in this corner, that is me outside the university on one of the really rare sunny days that we actually get in Scotland. And then also in my spare time, I like to travel, I like to go to concerts, I like to socialise, all of which have been pretty much curtailed by the COVID situation. But I do like to keep it in kind of science kind of nature when I go travelling. So here you have me in Brussels. So this is Atomium in Brussels and it's in Belgium. And if you ever get the opportunity to visit Belgium, I would highly recommend going, especially for everyone that's in science. It's pretty cool. So my research interests, as an analytical chemist, I feel like you either choose LC or you choose GC. So I am a big gas chromatography fan. I've worked with it throughout my projects. I've done GCFID, GCMS, and now on to 2D GC FeMS. And I'm in the Water Environmental Research Group at the University of Glasgow. So we're not all analytical chemists, we do have some microbiologists as well. So if you ever want to check out any of our research, that is our Twitter handle just there. So my research focuses mainly on environmental micropollutants, particularly pharmaceuticals and their trace analysis in sludge and biosolid samples. And for my transition, I would love to go on to some kind of postdoc, something in an environmental sector with a focus in an analytical chemistry, and hopefully with like a progression into a lectureship, because I've done some lab-based and teaching stuff with the undergrads and masters in my PhD, and it was pretty cool and I would like to do that. So just a bit of background about my research. So what are pharmaceutical micropollutants and why are they a problem? So when you take medications to help any ailments you have, so say you have a headache and you take some paracetamol or some Tylenol, then you fixes your headache and then you excrete it through feces and urine as either the unchanged pharmaceutical compound, the parent compound, or a metabolite. So it goes into the toilets and it goes through the sewer network and into the wastewater treatment plants with the idea that we can clean the water to a point that we can then use it again. However, the wastewater treatment plant processes, they're not suited to remove all the pharmaceuticals, and therefore they end up in the environment. So there's a lot of like treated wastewater and sludge samples that have a plethora of pharmaceutical compounds in it. And why is this a problem? Well, it has some detrimental effects on the environment, including feminization of fish with estrogens and the increase in antimicrobial resistance with any antibiotics. So my work focuses mainly on the treated sludge. So when it's treated, it removes all the harmful bacteria, but then they form these biosolid pellets that they can use in agriculture because they're quite nutrient rich and they use them as fertilizer. But if they're full of pharmaceuticals, then we're just throwing these back onto the land and they're going to leach and we're going to have a problem again. So my research is really into characterizing the pharmaceutical load in these biosolid samples. So I want to be as non-targeted as possible and have as little sample preparation as possible in order to see as many pharmaceuticals as we can, because we don't want to like exclude any with like an SPE treatment and all the acidic compounds go away or whatever. Now I can hear everyone being like, oh, why are you not using LC? But with LC, you can have a lot of solvent use because you're having your mobile phase, etc., and you're adding EDTA and stuff to chelate your ions. And then you have the matrix effects with LC. So we're choosing GC and hopefully making a method that's aligning with some green chemistry principles. So reduce solvent use, reduce energy consumption. But so my method, My research is basically into two parts. So we've got the optimization of the methods, being an extraction and derivatization, and then the application to the biosolid samples. So derivatization is the main thing you really need for GC analysis, particularly with polar compounds. So you need to increase the volatility, increase the thermal stability, and reduce the polarity of your compounds. So after a lot of reading, we decided that acylation was probably the best method to do so. And this is when you add a trimethyl silyl group to any active hydrogen containing compound. So you have an ease of acylation decreasing from your OH groups to your amides. And you really need a molar ratio of 2:1 with your MSTFA or your reagent to your active hydrogens. So I wanted to see which was best for our pharmaceuticals, and we decided to look at two acylation reagents which are of similar acylation potential. So MSTFA and BSTFA. And the BSTFA actually had 1% TMCS, which is a catalyst. So ideally it should have made it go quicker. And we looked at the acylation of paracetamol. So we've got our parent compound here. And then we have our acylation of our OH group to give us our mono-TMS formed, and then that silates again to give us our di-TMS group with our amide silating. And actually the amide seems to silate on your oxygen atom and not your nitrogen atom, as some people have thought, and we can confirm that using the mass spec, which is pretty cool. So our, as you can see here, the blue and the orange lines are your BSTFA. So we can see that the mono-TMS is still forming at first, and then it's going back down and your di-TMS is forming. Whereas with the MSTFA, which is the grey and the yellow lines, it's already reducing the mono-TMS and your di-TMS is forming. And this is with molar ratio. So we can see that MSTFA actually silates our paracetamol compound at a lower molar ratio. And so we thought this was ideal for our derivatization method. So then we looked at the effect of varying the molar ratio, like how much do we need to add to be able to silate these compounds. So we looked at carbamazepine this time, and it only monosilates on your amide group here. And we chose this because it was an amide. So as it's like one of the hardest groups to silate, we thought if we could get this to silate, then all the other groups should silate if they were easier compounds. So it doesn't actually go any further from the monosilation because of steric hindrance, so it doesn't actually allow any room for it to attack. It's a SN2 nucleophilic attack, so it won't give any room for the attack to take place. So we found that with increasing molar ratio, we actually see an increased relative response factor of our carbamazepine mono-TMS, and we see a reduction again in our carbamazepine parent compound with the highest molar ratio giving us the best RRF, because if you see here, the yellow, which is the lowest molar ratio, we don't actually get much reduction in our parent compound. So that suggested that we needed a higher molar ratio, so a higher volume of MSTFA. So we used design of experiment to optimize this. And then we used about 100 microliters of MSTFA, and we did this with both individual compounds and a pharmaceutical mix. So our individual compounds, each of them were chosen because they had varying number and type of functional group for the acylation mechanism. And then we did them individually and as a mixture. With the mixture, the RRFs were slightly lower than in an individual, but this is probably due to competing reactions because you've got more than one pharmaceutical competing for the MSTFA, so that's acceptable. And then the RSDs were all under 10, so we decided this method was acceptable for what we wanted to do. We then wanted to optimize our extraction method because we want to get these pharmaceuticals out as easy as possible. So we had the method online, and it was Gago, Ferrero et al, and we based it on that. So they, like them, they use 3 cycles of 2 ml methanol and milli-Q water at pH 2.5. So we ground our biosolid samples to increase the surface area, and then that'll lead for better diffusion. And we use a sonotrode device. So this is similar to your sonication probes and your sonication baths, but the sonotrode gives indirect sonication, and we use this little biopress so that you're not actually contaminating your sample because you're not putting the probe into it. So we sonicate it, and it like mixes it here, and then post-centrifuge, we centrifuge it, and then it gives us our supernatant. We do that for the 3 cycles. We collect all our supernatants and combine them. We then evaporate that off and we reconstitute it in ethyl acetate, ready for the derivatization step. So this process actually took about 40% less time, or it reduced our sample preparation time by 40% in comparison to the method that we based it on. So that was quite cool. So we then ran them on the GCMS, and we did actually see some pharmaceutical compounds. So we see ibuprofen mono TMS here, but as you can see, it's a tiny, tiny little peak. And there's all these other peaks around them, and they were interfering with the rest of the compounds we were expecting. And we just looked at the mass specs, and they were decided that they were fatty acids, but these are also derivatizable because they have their COOH groups. And the derivatized fatty acids were getting in the way. They were, we had run a pharmaceutical standard, and for example, the dodecanoic acid was coeluting with our metronidazole, and the hexadecanoic acid was coeluting with our ketoprofen standards, which is causing a bit of a problem. And like the EICs, the extracted ion chromatograms, weren't given as much either. And so instead of like having to go back and look at the extraction method again, because we did try and extract with as polar a solvent as possible to try and like not get these fatty acids in our extraction, we decided that we were going to use GC-GC top MS. So I'm not sure how like familiar people are with the concept, but it's basically like GCMS, except you have two columns. So you have your first column, which is generally your 30 to 60 meter column. And then you have a modulator followed by a second column, which is about 1 to 2 meters. And they're usually orthogonal. So you've got like polar and non-polar, or non-polar and then polar. And you separate first basically on boiling point and second on polarity. And then you get these lovely like 3D chromatograms with the peak intensity decided by your color. And this is ideal for complex mixtures because it has good separation efficiency, good peak capacity, and basically good for non-target analysis. So we looked at our biosolid samples that we extracted with our optimized method. And just to show you here, with our non-derivatized sample and our derivatized sample, our non-derivatized sample has a lot less peaks than our derivatized sample, and that's because obviously you're wanting to increase your polarity, increasing your thermostability and your volatility of the compounds with derivatization. So it just showed you that we definitely needed derivatization for the compounds. And again, we did see a pharmaceutical peak, and we managed to see salicylic acid di-TMS, and the mass spec at the top is the one that we actually got, and the one at the bottom is the one associated with the NIST library. So it came back saying like, yes, we have salicylic acid, and that was amazing. However, we didn't see many other pharmaceutical peaks, but I think this was to do with me running it at a split of 40:1, and ideally I should have really run it in splitless method, but I am confident if we did run it in splitless method, we would see a lot more peaks because we're seeing a lot lower limits of detection with the splitless method. But the other cool thing about your 2D chromatograms is the derivatized fatty acid peaks can be shown here. So all of these wee red dots are all the derivatized fatty acid peaks. And with this technique, they seem to be able to show like compound families of compounds. So you go up by like 1 carbon each time. And this is the same with over here. We had like glycerol and other sugars. And over here we had siloxane, which is something to do with the column, I assume. But yeah, so in conclusion, derivatization is required for analysis of pharmaceuticals on GC. A suitable method has been found for which derivatizes the majority of pharmaceuticals. The biosolid extraction method reduced sample preparation time by up to 40%. We can have a problem with fatty acid peaks on 1D GCMS, but ideally the 2D GCMS will overcome this because you're going on 2 different axes basically, provided that your split ratio is correct. But the thing I would take away from this is there is pharmaceutical compounds in the biosolid samples. So I just want to say a quick thank you to my team. So to Caroline, my supervisor, and to Celeste and Anka, and then also to Mark at the University of Strathclyde, who is running the GC-GC samples for us, and to the EPSRC, which is funding my research. And finally, thank you to FeMS for giving me the opportunity to showcase my research here. So with that, I'll take any questions. Thanks.
That was great, Kate. If anybody has any questions, go ahead and unmute, but definitely a big virtual round of applause. I'm going to throw one out there to get things going. So in your second to last slide, when you were showing your derivatized fatty acids, you showed most of them were lined up in the Y direction except for one. Is there a difference between saturation, or what was the difference in that one fatty acyl versus the other ones that were more retention time aligned?
I think it's probably something that's additional on the compound. They kind of go in a line like this, and I think you get the column bleed at the end as well, so it might go slightly further up. But yeah, usually you can see them dotted in a line, which is pretty cool.
Yeah, that was, and it's such a nice way to validate that that's what they are.
Yeah, I didn't know about the 2D GC before I started my PhD, but it's actually a really cool technique. And I know that was such a brief overview of it, but I would look into it if you have any opportunity to.
Yeah, no, that was very nice. Anybody else? If not, I've got another one for you. So warfarin's known to have lots of isomers. Did you guys look into isomers of warfarin? Because I saw that was on your list of one of your lists as just warfarin, but I know it has like 7 or 8 isomers, right?
I have not looked into any isomers at the moment. Warfarin was actually one that you definitely need derivatization to look at, but it's also really, really, really small in all my chromatograms, no matter what concentration I look at it in. But that might be an idea as to why. A lot of people in our research group can do the isotope different analysis, so maybe that would be something that I could try. So yeah, thank you for that.
Because I know it's really complicated. So if it works for separation in your method, that would be really cool.
Good idea. I'll keep that. Thank you.
Awesome. So let's give Kate a big round of applause, and we're going to go to Crystal now. So keep her on her toes.
Okay, can you see and also hear me?
Yep.
All right, cool. All right.
Looks
yeah,
great.
So I thought I was going to be last, but she's messing with me. But anyway, my name is Crystal Pace. I am a 4th year graduate student, or a rising 5th year, I guess now, since it's finally June. And I'm hoping to transition to industry coming up in the fall. So a brief history on me. I was born and raised mostly in North Carolina, in a very small town called Nashville, which gets very confused with Tennessee and also the Asheville Mountains. I moved to Raleigh to attend undergrad at Meredith College. And while I was there, my research focused on extracting natural dyes from fruits and vegetables for the purpose of dyeing fabrics and also creating dye-sensitized solar cells, which was really cool. But most of my time was spent characterizing those dyes using different analytical techniques. And that's what encouraged me and ignited a passion in analytical chemistry, and why I attended grad school. So I hopped across the street to NC State, which is literally across the street. And I'm studying and researching mass spectrometry under the advisement of Dr. David Munneman. Before I get into all those details, just some fun things. This is my family, my husband, Zach, that I've been with for 11 years now. And then this is our kitten, Ellie, who turns 4 tomorrow. So she's not really a kitten anymore, but she's still my baby. And other than my passion in the lab, I have a passion for ceramics and wheel-thrown pottery. So these are some fun things that I've made. And during the pandemic, I was able to start selling my pottery. So that's pretty cool. So now for the fun science, my research has focused on the development and application of a novel ionization source called IR-MALDESI, which stands for Infrared Matrix-Assisted Laser Desorption Electrospray Ionization. And as the name implies, it's a hybrid technique combining both MALDI as well as electrospray ionization platforms. And by doing that, we can get advantages from each of those soft ionization techniques. And so there are 4 primary components to IR-MALDESI. We have an electrospray ionization source, which operates like a typical electrospray. We have a mass spectrometer, and currently we have the Exploris 240, which is an Orbitrap instrument. So we are utilizing the high resolving power as well as accurate mass measurements. We have a translational stage that also has the capabilities of cooling for biological specimens. And then finally, we have an IR laser that operates at 2.94 microns. And that wavelength is specific to the IR absorption of water at that wavelength. And so this allows us to utilize the water in our endogenous water in our samples, as well as an exogenous ice matrix to act as an energy absorbing matrix to enhance desorption. And so By controlled exposure to humidity, we apply a thin ice layer on top of the tissue. And then we fire the laser at the tissue, which absorbs neutral species into the gas phase. And they interact with this orthogonal electrospray plume, which has charged droplets, and undergo an ESI-like mechanism. And so one of the biggest applications that we use IRML DESI for is mass spectrometry imaging. And this is really simple and just essentially entails taking a mass spectra at each location of a biological specimen. And we can take the abundances of each of these metabolites or lipids that we're detecting and build heat maps to show how an ion is changing across the surface of your sample. And so this is really important to understand how molecules are changing in biological samples, or even changing between control and or healthy and disease tissue sets. And so with this method, it requires minimal sample preparation. So in contrast to like an LC-MS run, where you have to homogenize your tissue and do some sample prep to isolate whatever molecules you want to analyze, we section our tissues and directly put them on the stage and start analyzing them. So there's almost no sample prep. And then we're able to get hundreds of analytes and monitor their abundances between different samples, and then also their spatial distribution. So you get a lot of information from just one experiment. So one of my first projects in my lab with IRML DESI was to investigate how flame retardants affect the spatial distributions of biochemicals in rat placenta. So flame retardants are pretty much applied to all of the furniture in your home, the walls, the building insulation, in order to slow down the ignition of fires, which is ideal. So that gives you more time to get out of your house. Unfortunately, though, they have been found to have really harmful biological and environmental effects. And for example, flame retardants have been detected at high levels in placenta tissue that gets donated to tissue banks. And so we know that it's making its way into biological tissues, and especially the placenta, which is an organ that exists for 9 months. So we're particularly interested in studying how the placenta is affected by these flame retardants, because it's the primary organ that's developed during pregnancy that is responsible for developing nutrients and facilitating the exchange to the developing fetus. So any disruption of that placenta can cause significant neurodevelopment effects. So specifically, we wanted to investigate neurotransmitters. However, this is a significant challenge in our field, the mass spec imaging field, because most techniques have this matrix interference effects that causes significant clusters in that M/Z range of the neurotransmitters. And that's due to a chemical matrix. So with IRML DESI, we don't utilize a chemical matrix. We only utilize ice, and that does not cause any interference effects. So we wanted to investigate how well we can detect neurotransmitters without having to do a bunch of chemical derivatization methods in order to optimize this for IRML DESI. So we first started by analyzing control placenta tissue, and we were able to detect 50 neurotransmitters and their related metabolites without chemical derivatization. And that's a significant amount, given that it's been limited to about 5 to 10 in a single experiment with other techniques. And so we then started to investigate their spatial distributions, and shown here is just one example of serotonin. And so shown on the right is controlled tissues, and we have both male and female placenta tissue. And as you go from right to left, you get higher exposure levels, and we observed that the abundance of serotonin was slightly decreasing at high exposure levels. And we noticed this in about 5 of their neurotransmitters. And so we investigated their pathways to see if we can find a cause for this, and we saw that it was likely in the production of this neurotransmitter, which could indicate that the enzyme activity is disrupted, which is something our collaborators had previously observed in their biological studies. Another project that I've been working on has been in the analysis of Inline Glycans. So inline glycans represent a post-translational modification on proteins, and they are polysaccharides and are incredibly complex molecules. And I say that very lightly, that much different from neurotransmitters. So there are many analytical challenges that they face, and these are just two examples that I think are relevant to my work. And so there's a monosaccharide called Cyalic Acid, which is this purple diamond that you see at the top of the glycan structure. It's known to be incredibly labile, and upon any additional internal energy, it dissociates from the glycan species and results in incorrect structures identified by Mass Spec. And so obviously that's not ideal. Additionally, that monosaccharide has a carboxylic acid or a negative charge, and that indicates a preference for negative mode ionization. And so both of these challenges have been reportedly reduced by electrospray ionization. And so because iron-Maldesi utilizes an ESI-like mechanism, we thought that iron-Maldesi could also be another ionization platform that would be very useful for the analysis of inline glycans. So to investigate this, I chose to study a well-known and well-sialated glycoprotein called bovine fetuin. And I prepped this using a filter-aided in glycone separation method, where you take the glycoprotein, you load it on a filter, a molecular weight cutoff filter, and you denature that protein and alkylate it. And then you cleave the in-link glycans from the protein using PNGSF. And then once they're cleaved, they can be eluted through the filter. And so the eluted glycans are then reconstituted and then directly pipetted onto our stage and analyzed very quickly. And so these are our primary results from this first analysis in negative mode ionization. And shown here are just all the structures that we detected in negative mode. And so all of these contain anywhere from 2 to 4 sialic acid residues, which is a significant amount given that we haven't done any chemical derivatization to stabilize those molecules. Additionally, we detected them at the second and third charge states, which is congruent with our ESI-like mechanism, as MALDI typically sees singly charged ions. And so my future work, and pretty much the last thing I'm working on before I graduate, is developing in-link glycan imaging methods in order to be able to visualize spatial distributions of these glycans in biological tissues. And so this is a lot more complicated than preparing glycoproteins or glycoprotein standards. And so we're working with Dr. Richard Drake and Dr. Peggy Angel to get this going with iron-Maldesi. And so, like I said at the beginning, I am hoping to transition into an industry position. I'm interested in research and development, method development, which I know is kind of vague, but I'm really open to many opportunities. I really do like mass spec, but I'm open to other analytical techniques as well. I am kind of rooted in North Carolina. So ideally, I would like to stay close by, somewhere in this mid-East Coast range. Hopefully I've been able to show you some of my research skills. So I just wanted to highlight some of my core values and something I'm looking for in a position. The first being personal and professional growth, which is vague, but what I mean by that is I'm a person that is constantly challenging myself and trying to learn new things. And so I want to be in a position where I'm running new methods or developing new methods and trying to answer different biological problems for our clients. And so I don't want to be in a position where we run the same method over and over for tons of samples, essentially. I enjoy collaborating with interdisciplinary teams. I think that science comes together, or science works when we all come together. And then I enjoy mentoring and providing direction, and also independently managing projects. So with that, I'd like to thank my research team and my advisor, Dr. David Mudeman, for his advisement during my research. And of course, my funding and my amazing collaborators who have really driven many of my projects. And finally, thanks to FeMS for giving me this opportunity to talk to you guys today about my research.
Awesome. Everybody give her a round of applause too, even though I did mess with you a little bit, Crystal.
It's okay.
But yeah, are there any questions? Everybody's being so quiet today.
So I have a question, well, kind of 1.5 questions. So my first half question is, what's a male placenta? Like, is it from a male pup or?
Yes, it
Okay.
corresponds to a male pup. Yeah, I
Okay.
forget. So trying to do this in like 10 minutes is super hard to get all the details. But yeah, so each pup has its own placenta. So we took a placenta from a male pup and also a female pup to see sex differences.
Okay. Like I was thinking, I can't be that ditzy. Like it must be the baby, right? So my real question is, your source, is that something you developed in-house or is it commercially available?
It's built in-house. We're working with a couple vendors right now to kind of try to commercialize it or get it in a way where it's ready to do that. But right now it's very much home built.
Cool. It's very cool. Thanks.
Yeah.
So I'll ask one. I always like asking Crystal questions. So
Practice for my pre-lit or for my
right?
final.
We'll ask you more philosophical this time. So you've been doing all these different imagings. I mean, you went from neurotransmitter to Inline Glycans. What would you really like to image that you haven't done yet?
Like molecule wise or
Yeah,
sample wise?
or both.
Well, I think for samples, I think it'd be really cool to look at like heart tissue, which I guess isn't truly my idea. I heard it once and I was like, that sounds really cool. We should do that. And no one's taken the initiative to get it done. So I'm like, it's still kind of like in the back of my head of like, that would be really cool to see, especially like the valves of the heart and stuff. But for molecules, I mean, we've done a lot of them. I've looked at proteins and peptides, and so I'm trying to think of what I haven't done in terms of different molecules, because I really have done.
Yeah, you've done a
I
plethora.
haven't shown it all to you because there's not enough time. But so yeah, I don't know. I think that glycans are still keeping me up at night. So
yeah, there's lots of work there. But yeah, awesome talk, Crystal. Everybody give her a big round of applause.
Thank you.
And we will head on to our third speaker. So great speakers today. So Ksenia, go ahead and get your slides ready to share.
Okay. Yes. Can you see it?
That looks great.
Okay. Good evening, everybody. I suppose it's not evening for most of you. My name is Ksenia. I don't know how to switch to the next slide. Okay, I know. I have graduated from Moscow State University, where I used to study nervous system of tardigrades. And shortly after graduation, I came to the laboratory of proteomics and protogenomics, where I started to study a totally different thing from me. And I was responsible for, over time, for pretty much everything from 2D geoelectrophoresis proteins and to sample preparation for mass spec and for data analysis from proteomic spectra. Recently, I came back to the university to study soil ecology, but since I was a biochemist by that time already, I was responsible for DNA barcoding of soil organisms, but that was part-time, and now I don't do it. I got some experience for DNA technologies though. And most recently, I got interested in cell biology also, and started to learn cell culturing techniques and other cell biology techniques. But besides all of my jobs, one of my most important occupation is that I'm a mother of a little girl. I really like talking about tardigrades, worms, and little girls, but we are supposed to talk about mass spec, so I'm going to give a quick overview of Proto-Genomic Approach that we are using in our laboratory, and a quick overview of my PhD project on chemical modification of cysteine residues, and a short overview of a project that I find the most interesting in our lab is proto-genomics of RNA editing products. So what is proto-genomic approach? Okay. Proto-genomic approach is developed to find protein products of single nucleotide substitutions. So first we take information from DNA or RNA and translate it to peptides. Then we add these peptides to a proteomic database of a particular organism taken from, for example, from UniProt. Then we search experimental mass spectra against this theoretical mass spectra from the database and extract the list of variant peptides bearing polymorphisms. The most challenging part of this approach is to validate our findings and make sure they're not false discoveries, as some of amino acid residues can go through chemical modifications over, for example, sample preparation. And for my PhD project, I have done research on chemical modification of methionine to isothreonine, which most likely happens during sample preparation as a result of alkylating of cysteine residues in order to destroy tertiary structure of proteins. We have tested various alkylating agents and found out that methionine to isothreonine conversion happens more often if the sample was treated with iodocetamide, which is the most commonly used alkylating agent. We also have tested these alkylating agents for overall performance in proteomic analysis and found out that iodocetamide yielded the lowest number of identified peptides, and chloroacetamide yielded the highest number of identified peptides. And common practice of quenching the excess of alkylating agent with the addition of the reducing agent after the reaction makes sense only for iodocetamide and doesn't seem to make sense for three others. Also, we have synthesized and analyzed analog peptides containing threonine and isothreonine in their sequences. It was done in order to try to distinguish between these isomers, because if we have threonine in the sequence, it's most likely came from the genomically encoded variation. And if we have isothreonine, it's most likely came from the modification of methionine to isothreonine. And we find out that B ion corresponding to the site of threonine in a peptide can be used as a diagnostical ion, as its relative intensity is significantly different in peptides peptides with isomeric amino acid residues. Really, it can be used, applied for whole proteomes only with the use of synthetic standards. And another project on proto-genomic of RNA editing. Sorry, I can't really see my slides myself. Okay, so RNA editing is post-transcriptional modification that is done by a specific enzyme. And sometimes it happens in codon regions of RNA. And when adenosine is getting edited to inosine, it is translated as guanosine. And if the region is coding, it results in a recoding of protein sequence. We have constructed the proteomic database based on transcriptomic data for fruit fly and searched proteomes of different developmental stages of the fruit fly against this database, and found out that the level of protein recording demonstrates stage-specific clustering. In other words, some proteins are expressed throughout all the ontogeny of the fruit fly, but get recorded at certain stages. And we can observe the so-called RNA editing blast at the late pupa and early adult stage of the fruit fly, and it was described on transcriptomic level, and now we can see it on proteomic level as well. The most intensively recorded proteins are involved in actin-myosin structure organization and physical trafficking, meaning that they function mostly in nervous system, and most of them, the most intensively recorded proteins are members of snare complex, which is responsible for physical fusion in nervous system. So mass spectrometry has a lot of applications in enormous number of fields in science. But being a biologist and a former zoologist, I'm mostly interested in mass spectrometry in biology and medicine, and looking for a postdoc position in this field. Okay, and I think that's it, and I will be happy to take questions.
That's a great talk.
Yeah, I am sorry, I'm a little confused with Zoom here.
Oh, no worries. Zoom is always
Which
confusing.
sounds strange because we've been using Zoom for so much, but okay, I can see you guys.
Nice. Any questions? So I've got one for you. I'm always full of questions. So your big false discovery problem, I'm sure, is huge, because if you get the wrong thing, so how do you really validate that and make sure that you aren't getting any or aren't giving out any false information or finding false hits?
Yeah, I had to drop a lot of details, and we've actually been through several different projects on proto-genomics, and we solve this problem differently from time to time, but the most common way is to just synthesize the peptides of interest and do MRM analysis and make sure, measure them and make sure they are actually in the sample, which is usually asked by all of the journals to do, and we always do that. But on the other hand, you validate mass spectrometry with mass spectrometry, which is a little like, it's anyway, like when you do MRM analysis, it is much more sensitive than shotgun proteomics. So you, of course, you can get more confidence. But recently, for the work on the fruit fly, we did some bioinformatic analysis as those peptides that were selected as variant peptides. They were also analyzed with special tools of retention time prediction, and like to get rid of all outliers or false discoveries. They had some bioinformatic tools for that also. Or, for example, the retention time prediction, and you compare whether the retention time of your findings are actually normal and not completely different from the theoretical retention time of the ions that you see. And also, since we don't have a lot of peptides in our list, finally, we can just open up spectra and just manually check that the spectra of our findings are looking adequate and not something completely different. So usually it ends up with manual correction of the spectra as well.
Yeah, no, that makes sense. Anybody else? Well, if nothing else, let's give a huge round of applause to all of our speakers. Those were great talks today. So I learned a lot from everybody, and everybody's working in such different areas. That was really cool. And so we'll open up for breakout rooms now. Does anybody have any announcements before we go?
I could just jump in to say that abstract submission is open for MSACL Europe in September. It'll be virtual. So if you have anything clinically related, we would be happy to receive abstracts. And also BASA Lab showcase is coming up in about an hour. So I'd love to see you there to learn more about Carrie Basa's science and the other members of her lab. That's on msacl.org.
Nice. Anything else? We don't have our next FeMS session set yet, so we'll keep you guys updated on that. But I've been a little slow, so we don't have anything for July, but we'll get something. But yeah, so everybody have a great day if you have to log off. If not, I hope you can stick around and get to meet some people from around the world and visit about your science. But I'll start the breakout rooms now.
I think we might be few enough that we could be in one.
I know, that's true. Yeah, I might split into two, and then so I'll split into two so you guys can meet more. And if people drop off, then I'll just combine everybody. But I'll see you guys, and stay on as long as you want. I'll just keep this open.